afternoon, everyone. I think it is afternoon now, yes. And my name is Dr. Chad Keates. I am a herpetologist and a molecular biologist that up until this point has been very much focused in the taxonomical field, systematics. I've done quite a bit of research in Southern Africa, Angola, Zambia, worked quite extensively with people like Vanna Conradi and many of the other herpetologists. And I was brought into this team to have a look at some of the frogs that we're seeing up in this Khakea Bray Transboundary system. And I was very, very excited to, because obviously this place has not been studied very well. It's a very understudied part of the region. Northwest in general is not a very well studied area. So I was quite excited to get in there. Great stuff. So to get us started, the KBTA plays host to a high diversity. As my colleagues have alluded to, this area is a very interesting place for invertebrates, vertebrates, and fauna and flora. And frogs are very key components of this diversity. Frogs are one of the most important vertebrate taxa on planet Earth. They are phenomenal ecological indicators, and they're a good sign of the health of ecosystems. Often you find different assemblages of diversity based on the toxicity of the water. If you have certain kind of conditions, you get certain species, and some species dominate in other systems. So just knowing what frogs are around and the diversity and the density of these areas can really tell you a lot about the health of these systems for humans and for wildlife. And also frogs are phenomenal ecological indicators. I just said that, sorry, and phenomenal ecosystem contributors. So they actually contribute to both as the prey item, as the predator as well, and they export pressure to the terrestrial environment post metamorphization. So as everyone knows, many of our frog species obviously grow up in the water source. They come as tadpoles, move into froglets, and then as they move out the water, they become fully fledged frogs. But while in the water, they often become prey to many types of animals, so fish, many invertebrates, and therefore they are contributing to the lower levels of the food chain. And as they move out, they become both predator and prey to terrestrial environments. So they're a very, very important part of our ecosystems, and understanding them really helps us understand what's going on in systems and if we need to intervene or if we need to conserve certain section of the wildlife. So the frogs of Khakea Bray, if you have to go into Louis de Prere's recent copy of Frogs of Southern Africa, you will find these, I think it's 12 frog species. There isn't that many frogs found there, but the ones we do get there are quite common. They've written some interesting ones, such as the one pictured there, which is your banded river frog, Phrynomantis bifasciatus, the beautiful species. Among other species here, such as the giant bullfrog, which Tatena just spoke about. Also, we have the frogs of Khakea from the ADU perspective, so the animal demographic units. This is a mapping service where citizen scientists can log their records online, and we can have an understanding of what species are found where using quarter-degree cells. And what's so interesting to note is that the olive toad is found on the animal demographic units, but it's not found in Louis de Prere's book. Whilst the red toad is regarded as being present in the area in Louis de Prere's book, but it's actually absent from the ADU. So there's a bit of discrepancy between what species are found there and which ones aren't, which leads into the motivation for this project, which is really to have a look at this area, see what is there, and just improve our understanding of the frog assemblages of this region. Because as we can see from the ADU and the frog book by Louis de Prere, there is a discrepancy between the sources, which means we don't actually have a handle of what's going on. And this is interesting because most of the rest of the country, there are phenomenal resources, especially with the frog world, in terms of what we know is there. So if you go to KZN, Gauteng, Western Cape, Eastern Cape, we have a good understanding of what's present there. Many of the quarter-degree cells are filled, but in Northwest, there's a lot of vacant QDS cells. And another part of this is that there was a lovely paper done by Bots in 2011. And this actually highlights the influence that cities, roads, and protected areas have on our understanding of frog diversity and endemism in the country. So this is obviously a map of one of the species in the country, but as you can see with the circle near Tosca, there's very few records out there. And this seems to be the same for all the species that are found in the region, is that it's a very undersampled part of the world. And the reason for this that many people believe is that it's a sampling bias. So there's no major cities, there's no major road networks that pass through this region. So we don't have many citizen scientists going into these areas and logging these records, which is which makes it a very priority area for us to move into, because we don't actually know the true diversity of this area. And one of the good examples of this is this skink here, which is the Panaspis warburgi, which is your Warburg snake-eyed skink. It's quite a common species of skink that you find along the coast, up from Eastern Cape through KZN to Mozambique. And when I was there in this last trip, it was quite dry, but I was looking under rocks, obviously, for reptiles as well. And I managed to find this skink with very minimal effort. It took me 5 minutes, flipped this rock, and I found this little guy 20 kilometers west of Tosca. And this represented a plus 50-kilometer range extension for this animal. And that took, I can say, like 5 to 10 minutes of looking. And I can't imagine how much we will start to find with reptiles, frogs, even scorpions and spiders if we just look there for a little bit longer. In addition to that, I also got Nucris intertexta, which is your strum, I can't remember the common name for it, sorry. And then you get Pedioplanus lineosalata, which is your spotted sand lizard, the Pachydaktylus capensis, which is your spotted thick-toed gecko. All of these represented new findings for their quarter-degree cells when I was there. So I was finding reptiles the whole time I was there with very little effort, actually. I was looking on the side while looking at the pans, and I've managed to find these little guys. And what's so interesting about it is that if we had to look a bit longer, we'd find a lot more other things. And to put in perspective, you've got a few species, about 10 species of frog here, listed on the screen. And these are their current distributions according to the animal demographic units. So southern pygmy toad, northern pygmy toad, raucous toad, snoring puddle frog, plain grass frog, broad-banded grass frog, Poynton's river frog, striped stream frog, Natal sand frog, and then your foam nest frog. And with a bit of increased sampling effort in the Tosca region and the Khakea Bray transfrontier aquifer system, I believe that species like this might start cropping up, and our diversity will increase from an approximate 13 species to maybe 14 or 15. And there's no telling if maybe some of these species not found in the Northwest will start being found there too. So this is a little bit of a humorous one from some of the team, but I believe increased sampling in the area will definitely result in the discovery of several of these species in the KBTA. And if not, several samples will definitely be interesting to look into, because of obviously in this country right now, there's a lot of work going into genetic analysis, especially in regards to species, uploading them to the BOLT system and determining the p-distance analysis, or with p-distance analysis, with the distance between some of these taxa. And cryptic speciation is a very, very big part of frog diversity in Southern Africa. So having a look at some of these samples under a new light would definitely be something to look at. Also, an unimportant species, as Tatenda alluded to, is this giant bullfrog, Pixicebulus adspersus. It's an important species both because it's the largest frog in Southern Africa and because it's a flash breeder. So you find them quite extensively in the Khakea Bray region, and this is because they use these pans. They breed quite quickly, and when it's done, they will actually burrow in the soil or the mud up to 1 meter deep. And as much as this species is not threatened right now in parts of Southern Africa, especially parts of Zimbabwe, they are being harvested for food, and they're starting to disappear from parts of the country that they used to be found. They're a species that we definitely protect because they're a very important part of the ecosystem. They're larger frogs, so they form prey for a lot more animals, and they actually are predators eating reptiles, birds, and small mice. In addition to that, they're a key part of most wetland systems in the areas they actually exist in. So having a better understanding of this species, where they are, and how they are doing is a very, very important part of this project. So the main aim of this project is to determine the diversity of the frogs in the area and to see exactly what is around. So the methods for this study are quite broad. So a lot of it's going to be active searching and netting. So for frog ecology, or frog herping, for herping, what we tend to do is we tend to look around in the area near the wetland system, lift up logs, lift up rocks, and see what we can find. We also use a netting where we throw the nets into the water and we pull out the tadpoles and identify them to species level. In addition to this, we visit each site at night, because if we're looking in the day, sometimes we're not going to find some of these cryptic taxa that are only using the nighttime to breed. So that's an important part of this, is to look at day and at night in each system. Each amphibian will be identified at each site, and we'll take isotope analysis. So for this study, there will be this kind of analysis done. So we'll take 5 fin or tail clips per species per pan. So The amount of pans is yet to be determined, but there's quite a few of them. So it'll be very interesting to see the difference between the different pans, between the different species. And for genetics, we will take 5 toe clips, or we'll take specimens and preserve them, depending on what is required for this project. And we will preserve them in 97% ethanol. These samples will be amplified using the genes that are most commonly available on GenBank, and then we will use this to determine if there's cryptic speciation in the area, and what's the population dynamics of the area. So for population genetics, grabbing a few samples per pan per species is really going to show an interesting tail. And if there's something that we believe may represent undiscovered taxa or is a range extension of note, we will be taking voucher specimens, and these will be accessioned most likely at the Port Elizabeth Museum in Baywald. The potential project benefits, like I've listed before, are absolutely numerous. It's a completely understudied part of the world, and understudied parts of the country, actually. There's not many left. So to have a look at what's there, especially in the frog world, is extremely exciting, both as a scientist and just an amateur or professional herpetologist. Also, we want to determine if there's sensitive species, and also frogs will obviously indicators of the health of these systems. So determining the diversity will tell us what's going on between the different pans. We also look at Pixocephalus and other important species and develop baseline data for all the other genetic projects. So in this country right now, there's quite a few projects happening on species like Cacosternum, which are your cacos, your river frogs, and your stream frogs. So samples from this study will definitely feed into other papers, which will mean that there's a huge amount of scientific exposure for this project. And species lists will be developed for the area, as well as we believe several publications, especially in the biodiversity of the area. So it was a bit of a short talk, but my colleagues captured the ecology of the area quite well. Thank you so much.